Hi everyone, it's Dr. V. I am back talking about more microbiology related topics. I am going to talk about just the basic anatomy of the bacteria. Not going too detailed, but this will be a good review of general bacterial structures. Just as a reminder, I'm sure you remember this maybe from previous classes that you've taken, but microbiology, microorganisms are divided according to eukaryotic and prokaryotic. The prokaryotes are generally the bacteria. Then we have some other eukaryotic microorganisms that includes like protozoans and also fungi. But one thing hopefully you remember from previous classes is that eukaryotes have a membrane, okay? Around the nucleus. So they have a membrane-bound nucleus. They also have mitochondria, which is a double membrane-bound organelle, as well as an endoplasmic reticulum. Now, there may be some modifications to some of these structures, like the ER in the lower eukaryotes, but prokaryotic organisms do not have a nucleus. We'll talk about it. They have what's called a nucleoid. All right, so here, before I talk about the structure, I do want to just review really quickly the shapes of bacteria and some of the arrangements. The circular shaped bacteria are called cocci. Okay, so we see it here. They can be in different arrangements. If they're in a set of two, we call it diplococci. If they're in clusters, we call them staphylococci. In chains, we call it strep. And we have sarcinae and tetrads. Sarcinaes are 8, tetrads are arranged in 4. So the circular shape is called cocci. The bacillus is more rod-shaped. Okay, so we see it here. So if we see rods, we refer them to bacilli. Some of them can kind of be ovalish in nature, so like extremely short rods, we call those coccobacilli. And just like we saw diplococci for the circular, we have diplobacilli for the set of 2, streptobacilli for the chains. So bacilli is the term that we refer to for the rods, where cocci are the circles. We do have some bacteria that have some extensions that grow out like hyphae and stalks, but we do see that a little bit more, especially those hyphae in the fungi. And here are some other ones here, but I just want to highlight there's some bacteria that has a filamentous structure, and we have the spirochetes, which are more corkscrew shaped, and they are very flexible in nature. And then we have the spirillum, which are like helical in nature, but they are very rigid in structure. All right, so we'll talk a little bit more about the bacterial structures. All right, so here is showing you the basic anatomy of the bacteria. We have attachment structures. Attachment structures includes like the pilus, some people may call it pilus, or the fimbriae, which are in the same family. One thing we have to remember is that when there's a pathogen that is trying to invade the host or get inside the host, the first thing they must do is actually attach to their host, okay? So if they can attach, then they can launch, their invasion. So those structures are very important for that. On the outside of some bacteria, they may have a structure called a capsule. The capsule tends to be pathogenic, and I'll explain that in a second. The bacterial cell wall is actually a critical component for classes of bacteria. We have some organisms that have a really thick cell wall, which are the gram-positives, and those that have very thin cell walls, those are the gram-negatives. I do have a video that I will be posting shortly on the cell wall, so make sure that you hit that bell so you'll get notification when I submit it. Bacteria also have plasma membranes, very similar to what we have. They have a region where their DNA is found called a nucleoid. They have ribosomes that are involved in translation, and of course they have cytoplasm. So I know I'm kind of rushing this because I'm actually going to talk about each one of these structures. So the first thing I'm going to start with is the region of the bacteria where you find their DNA or nucleic acid. That region is called the nucleoid. So we see that our DNA are found in here. That is the region called the nucleoid. Prokaryotes have that. Now here is a structure that we see in the gram negatives. Now if you don't know what I'm talking about, there will be a video coming out that's talking about comparing and contrasting the gram positive and the gram negative. So you'd be able to see a little bit more what I'm talking about. But the gram negative has this structure called the lipopolysaccharide, also known as LPS. So if you want to break it down, lipo refers to lipid, poly means many, saccharide means sugar. So lipopolysaccharides are composed of lipid and sugars. So I want to mention to you, before I show you the actual picture close up, the lipid portion is called lipid A. All right, so I'll talk about that. So here's lipid A, here's lipid A. Lipid A is what we call a super antigen. So if your immune system has exposure to it, what will happen is that your immune system will be extremely hyper stimulated. And if that happens, that can lead to a systemic inflammatory response that can actually lead to shock and even death of the person that is infected with a gram negative that has these levels of LPS. So let's take a look at its structure really quickly. Here is the lipid A, that's the lipid portion. This part is anchored into the bacteria itself, so it's really typically not exposed to the environment unless the organism is dead. If the organism dies, then we know that they burst open, and that's when the lipid A can have access to the host immune system. But typically is embedded inside of the bacteria. Now, sticking out of the bacteria, we have the core sugars. You see each one of these represent a sugar, and then we have our outer structure, the outer sugars, which are known as the O antigen, and they can vary. Now the O antigen can act as a fingerprint to identify the bacteria. So if there's let's say a diarrheal outbreak and they cultured a certain strain of E. coli, they can match the O type to see what it is. The E. coli strain that has been the worst one is E. coli 0157. The O comes from the O antigen. Now let's take a look at the colorful picture. I think it's really nice. Here's showing you the lipid A, that's the lipid portion of the LPS. Remember, that is the bad part of it in terms of us. If we get infected with it, it can actually cause a lot of issues. Then we have our core sugars and then the O antigens. This is the part that's sticking outside of the cell. This is the part that's inside of the membrane of the bacteria. Collectively, the sugar part of LPS does not pose a major problem. It is the lipid A portion that can elicit a shock response. Not good. See this face right here? That is not good. Okay, so here are some structures that are also found in bacteria. They're called inclusion bodies. Inclusion bodies are more like vesicles that they store things. And if you take a look here, you see that there are these little storage regions right here. Those are inclusion bodies, like vesicles for storage. And they can store various things. They can store carboxysomes, gas, iron. There's a lot of different things that it can store. So just to let you know that they are called inclusion bodies. All right, so let's talk about DNA, bacterial DNA and plasmids. First, to start off with, hopefully you remember from previous classes that the DNA found in eukaryotes, like you, your dog, your plants, they are linear in nature. And we tend to have more than one chromosome. Like humans have 23 sets, right? Or 46 chromosomes. Bacteria only has one, okay? One chromosome only. See, it says one right here. It is circular in nature, even though it can coil up. Okay, so we can see here there is one that is coiled, and there are mechanisms that they use to cause it to coil and supercoil. Now, the one chromosome that they have encodes important genetic information. So everything they need to survive is found on that chromosome. However, bacteria have also extra pieces of chromosome called plasmids. There's different types of plasmids. They do not encode anything really essential for life, but they can encode for other things like drug resistance, like antibiotic resistance. We call those type of plasmids R plasmids, R for resistance. Okay, we have some that have virulent genes on it, or genes that code for things that could be harmful. So they're not really needed for life, but they are needed to maybe enhance the survival of the organism or allow the organism to be more pathogenic. All right, so now we're moving to the actual attachment structures. So remember I said there were two structures that are needed for attachment. The first one is fimbriae and the other one was pili. So if you take a look at this, these extensions that are coming out of the bacteria, those are known as fimbriae. A lot of times organisms, especially those that attach to epithelium, actually need these structures. Most bacteria, especially those gram-negatives, have it. They're not, and if they don't have it, they can use other things to adhere, like adhesion proteins. But typically the fimbriae are important for that. The other attachment structure are pili, but I want to actually just zoom a little bit in on a particular type of pili called the sex pili. So here we see an actual bacteria that has a plasmid in it. You remember, plasmids are extra pieces of chromosomes. Not essential for survival, but they aid in other things. So here's the F plasmid that plays a role in a process called conjugation. So what it does, we will have an extension of sex pili that will then attach to a recipient. So the organism that has the F plasmid is known as the donor. Okay, and what it's going to donate is an actual copy of the plasmid. Now here's the kicker. All right, so the plasmid goes through that pili. So after the process, that donor, we call that an F positive cell, will then convert the recipient into a donor. Okay, so it then also becomes the F, the F plus cell, because it will now have the plasmid in it. This process is actually called conjugation. Okay, but just to let you know, as an example of an attachment structure of a pili, the sex pili is involved in conjugation, and the donor typically has the F plasmid, and once they transfer it, the recipient becomes F positive. What a transformation, huh? Here we're going to talk about a very important structure, and that is the bacterial capsule. Now the capsule, I tell my students this all the time, the bacterial capsule, I like to think of it as a slippery soap. You're like, what in the world does she mean by slippery soap? So if you ever put a bar soap in the water and you let it sit, you notice that the bar soap gets kind of slimy and nasty on the outside. And if you were to try to pick up the soap, it will slip out of your hand. The bacterial capsule is kind of like that slippery soap. So if you take a look here, this is a fluorescent staining of the capsule that surrounds the bacteria. A lot of pathogenic bacteria has this. This particular organism, Bacillus anthracis, actually causes anthrax. So if we take a look here, if a phagocytic cell try to come along and engulf it. You remember, it's like the slippery soap. It will slip out of your hand if it was you trying to pick up a soap. But if a phagocyte tries to eat this, try to engulf this, the capsule actually prevents the white blood cell from engulfing it. So it's anti-phagocytic in nature. So it's resistant to phagocytosis. So the bacterial capsule is very critical for pathogens, especially if you want a pathogen to escape the immune system. This is one of the best ways to do it, because if the phagocyte can't engulf it, it will just go ahead and spread all around the body or go wherever it needs to go and causes issues. Okay, so remember the bacterial capsule, not all bacteria have it. The pathogenic ones, a lot of them do have it. Not all of them, but a lot of them do. So it's an important structure for certain pathogens. All right, just like how humans, like males, have sperm cells with flagella that helps it to swim, bacteria also has flagella for locomotion. Now the composition is a little bit different. Like the flagella in bacteria actually is composed of flagellin. But I just want to show you that bacteria has different arrangements of the flagella. Once again, it's all for movement. If a bacteria needs to swim towards an area with oxygen, it will use its receptor, detect where there's oxygen or where there's a lack of oxygen, if there's a bacteria that doesn't like oxygen. And it will swim to that region using a process called chemotaxis. But bacteria that has one flagella is called a monotrichous flagella arrangement bacteria. Then bacteria that has two, one on each end, is called amphitrichous. Bacteria that has multiple flagella on one end, or you may see multiple on both ends, that's termed lophotrichous. And peritrichous means that you'll have flagella surrounding the organism pretty much all the way around. So these are the four different arrangements. Certain bacteria may have none, some may have one, some may have a lot, so many different arrangements, but its entire purpose is for locomotion. Okay, so let's go ahead and let's take a look and see what you recall from the lecture about the basic structure of. All right, first question. Which organisms have lipopolysaccharide? And just in case you're not remembering what that big word means, it's also known as LPS. So which one? Gram-positive, negative, or both? Hopefully you picked gram-negative organisms. Remember, I said the gram-negative has the LPS. If you don't understand what gram-positive versus gram-negative is, please make sure to look out for that video that's coming out very, very soon on me describing the cell envelope between the gram-positive and the gram-negative. Next question. Which portion of the LPS is considered toxic? So remember, the LPS has three structures: lipid A, O polysaccharides, core polysaccharides. Now, which portion is toxic that can lead to shock? Let's see if you remember. That is lipid A. Remember, lipid A is a super antigen. So what it will do is elicit a very strong inflammatory response if it has access to the immune system, and that can actually be the demise of the host, okay? All right, next question. Which bacterial structure plays a major role in blocking phagocytosis? Is it the capsule, LPS, inclusion bodies, plasmids, or cell wall? Let's see. If you picked capsule, you are 100% correct. Okay, so remember, the capsule is the slippery soap. Okay, just imagine trying to pick up that slippery soap that's been sitting in water. That will not happen. Next question. Which flagella arrangement is described by having multiple flagella on one or both ends? The key is multiple flagella on one or both ends. Is it peritrichous, lophotrichous, amphitrichous, monotrichous, or none of the above? Just look at those prefixes. If you picked lophotrichous, you are correct. All right, next one. The region where the bacterial DNA is present is called, is it nucleus, nucleoid, nuclear, membrane, or none of the above? Now this one, hopefully you get it. The answer is nucleoid. Okay, so this was a very brief review over bacterial anatomy to make sure you know the basic anatomy of the prokaryote. Hopefully you got 100%. These concepts are very fundamental and they build on throughout the course if you're taking microbiology. So please make sure you understand this. If you did not get 100%, which I hope you all did, please go back and watch the video again from the beginning so you can kind of gain an understanding. Please leave a comment. Let me know what you learned. Let me know if you got 100, or if you have any questions regarding any of the information that was discussed in this topic. Please let me know, and I will definitely get a response to you. Take care. Bye.